now I can start.
Yes.
Okay, good morning, and thank you for inviting me to give this presentation about epigenetics in human obesity and type 2 diabetes. So you all probably know type 2 diabetes is a multifactorial disease, and it's well established that combinations of genetic and non-genetic factors such as aging, diet and obesity, physical inactivity, and intrauterine environment all affect the disease. And data from my group and others also support that epigenetic mechanisms have a key role in the pathogenesis of type 2 diabetes. So we have an epigenome, and it includes DNA methylation, histone modifications, and non-coding RNA. And my group has mainly been studying DNA methylation, although more recent studies also include non-coding RNAs and histone modifications. So we have an epigenome, and it controls cell and tissue-specific gene expression, X chromosome inactivation, parental imprinting, and transcriptional control during cell differentiation. And dysregulation of epigenetic mechanisms may contribute to disease. So when we started this work approximately 18 years ago, we asked the question, do epigenetic alterations play a role in the pathogenesis of type 2 diabetes? And to answer this question, we used and worked on the following model, where first we examine if there are epigenetic modifications in tissues from patients with type 2 diabetes versus controls. However, we identified several of those epigenetic differences, but when you find a difference in a diseased versus control person, you cannot tell whether this modification contributed to that you got the disease, or whether it potentially occurred after you received or got the disease. So therefore, we have also studied the effect of non-genetic risk factors for type 2 diabetes, such as aging, an impaired intrauterine environment, diet and obesity, and the effect of physical activity. And there is also a strong link between genetic factors and the epigenome. So we therefore have done so-called MQTL studies, where we have linked genome-wide SNP analysis to the DNA methylation pattern and linked the genetic factors to the epigenome and thereby diabetes. We have written a few review papers. One of the more recent one was published in Cell Metabolism, where we summarized data from our group and others. More recently, we have tried to test whether we can use our epigenetic findings for patients and perform translational studies. And to do this, we have performed genome-wide epigenetic analysis in humans to discover causal epigenetic changes and potential epigenetic biomarkers that can be used for prediction, prevention, and treatment of diabetes. And today I will present one such study, which is the first study, and I will also present two other studies, one linked to obesity and one to type 2 diabetes. So the first study I want to present data from was published in Science Translational Medicine approximately a year ago. And here our goal was to find blood-based epigenetic biomarkers that can predict whether drug-naive, newly diagnosed patients with type 2 diabetes will respond to and tolerate metformin, or whether they are non-responders and or intolerant to metformin. So metformin is recommended as the first-line treatment for type 2 diabetes, and globally over 150 million people are prescribed this drug annually. However, approximately 30% of subjects fail metformin therapy, and approximately 20 to 30% experience intolerable side effects. Optimal treatment of diabetic patients is crucial because a poor glycemic control increases mortality in diabetic patients. and identifying those patients more likely to fail or reject metformin would aid clinical decision making. Currently, there are no phenotypes or biomarkers that successfully predict the glycemic response to or tolerance of metformin in newly diagnosed patients with diabetes. So we designed the following study where we had newly diagnosed drug-naive patients with type 2 diabetes, and we had blood samples taken before they started their treatment. And then all of them went on monotherapy with metformin, and we identified after approximately 1 year the ones that had responded to the therapy with a reduction in HbA1c according to ADA and ESD guidelines. We also identified subjects who were non-responders who did not reduce their blood glucose based on HbA1c measurements. We also identified subjects who were tolerant and intolerant to metformin based on gastrointestinal severe side effects. And in these subjects, we analyzed DNA methylation baseline, so before they started the metformin treatment in blood, using the Epigenetic, which is a genome-wide analysis for DNA methylation. And we did this in 3 different cohorts. One, the Undis Discovery cohort, which is a cohort from the south of Sweden. And we also split this cohort into 2. So we have the Undis replication cohort. And then we also use the European replication cohort. And this includes samples from Latvia, from the OptiMed study, and also samples from UNDU, which is a cohort in Uppsala. And we found that DNA methylation of 33 CpG sites were associated with response to metformin in all 3 cohorts. We then meta-analyzed these results and performed a Bonferroni correction. And after the Bonferroni correction, we found that DNA methylation of 11 CpG sites was associated with future response to metformin in patients with diabetes. And regarding intolerance, we found that DNA methylation of 7 CpG sites was associated with future metformin intolerance in all 3 cohorts. And after a meta-analysis and Bonferroni correction, 4 of these CpG sites stood correction for multiple testing. We then found that performing a logistic regression model, that DNA methylation, that increased DNA methylation of all these 11 CpG sites was associated with an increased risk of not responding to metformin, while increased DNA methylation of the 4 CpG sites that we had identified was associated with an increased risk of not tolerating metformin. We then generated a methylation risk score for the 11 CpG sites where methylation was associated with a response to therapy. And as you can see here, in both replication cohorts, there was an increased, the methylation risk score was increased in the non-responders compared to the responders. And we then generated area under the curve and found that this methylation risk score could clearly discriminate between responders and non-responders with AUC of 0.80 and 0.89 in the replication cohorts. We did the same for the sites which was associated with intolerance. We generated the methylation risk score for these 4 CpG sites. And as you can see here, this methylation risk score was higher in subjects who were intolerant to metformin in both replication cohorts. And this methylation risk score could clearly discriminate between tolerant and intolerant subjects to metformin with AUCs of 0.94 and 0.89 in the replication cohorts. We then tested whether clinical variables also could discriminate between responders and non-responders and the tolerant and intolerant individuals. But as you can see here, none of these clinical variables gave a good AUC. So to summarize this study, we have analyzed DNA methylation in blood from newly diagnosed drug-naive subjects with type 2 diabetes. And we have identified DNA methylation that can discriminate responders from non-responders and tolerant from intolerant subjects to metformin. And we will now develop these biomarkers further to see if these can be used to decide if people should get other anti-diabetic medications at baseline and thereby reduce vascular complications and mortality and suffering for the patients. So the second study I want to present was published in Diabetes earlier this year, and the aim with this study was to investigate if a lifestyle intervention in pregnant mothers with obesity was associated with epigenetic alterations in cord blood and body composition in the offspring. So in this study, pregnant mothers with obesity were randomized into a lifestyle intervention that either included physical activity and a diet intervention, or they were randomized into a control group. And then we analyzed DNA methylation using the 450K in cord blood of the offspring. And we identified that DNA methylation of 379 sites were different in cord blood of the offspring of mothers who have been included in the lifestyle intervention. And these were annotated to 370 genes, and performing pathway analysis of these genes, we found that there was an overrepresentation of genes annotated to response to fatty acids and adipose tissue development. We then performed a causal mediation analysis of these 379, or methylation of these 379 sites, and could show that DNA methylation of 17 sites was associated with or seemed to cause the lean mass in the offspring. And we could also show that the lean mass in the subjects of mothers included in the intervention had an increased lean mass. So to summarize this study, our study demonstrates that lifestyle interventions in pregnant women with obesity are associated with epigenetic changes and increased lean mass in the offspring. So the third study that I want to present data from was published in Nature Communications earlier this year. And the aim of this study was to explore whether alterations in muscle stem cells from individuals with type 2 diabetes affect muscle regeneration and glucose uptake. So in this study, muscle stem cells were collected from subjects with type 2 diabetes and control individuals. And then these muscle stem cells, called myoblasts, were differentiated into myotubes, and we analyzed DNA methylation and gene expression genome-wide, both in the myoblasts and in the myotubes. And we found that during differentiation, more than twice as many DNA methylation changes took place during differentiation of myoblasts into myotubes from individuals with type 2 diabetes compared with the controls. So we found that when myoblasts from the diabetic people differentiated into myotubes, there were more than 100,000 changes in DNA methylation, while when myoblasts from the controls differentiated into myotubes, only 50,000 changes took place. So since these cells were cultured in vitro in the same condition, it suggests that there is an epigenetic memory from the diabetic in vivo situation in the cells taken from diabetic people. We then wanted to identify new or novel candidate genes that may contribute to muscle phenotypes seen in diabetic people, where the muscle may have a lower quality and a lower strength, and there is insulin resistance. So looking into the gene expression data, we identified a gene with the name VPS39, where we found that both in myoblasts and in the myotubes, there was a lower gene expression in the cells from diabetic compared to control people. We also found that there was a negative correlation between VPS39 expression and DNA methylation in the muscle cells. And to test whether actually DNA methylation directly affected VPS39 expression in the myoblasts, we cloned the promoter of VPS39 and methylated that in vitro and then performed a luciferase assay. And as you can see here, increased DNA methylation of the VPS39 promoter resulted in a reduced transcriptional activity, supporting that there is a direct effect of methylation on regulation of VPS39. We then wanted to understand how VPS39 or what it does in the muscle cells. And since we had lower expression in both myoblasts and myotubes from diabetic people, we silenced VPS39 in myoblasts and then differentiated them out into myotubes in vitro. And what happened then was that we got lower number of myotubes. So there was a clear reduction in the cell's capacity to make myotubes when we had lower VPS39. And this can be seen also in this figure. So here we have myoblasts, and this is in the control. This can clearly differentiate into myotubes, but when we silence VPS39, the cells can no longer differentiate into myotubes, supporting the VPS39 is important for muscle regeneration. And to better understand what is going on when we have reduced VPS39, we analyzed gene expression genome-wide in myoblasts where VPS39 had been silenced and in the controls. And we then found that genes regulating muscle function were reduced. We also found that many muscle-specific genes had reduced expression in VPS-silenced muscle cells. We found that genes regulating cell proliferation was upregulated. And we found that genes involved in autophagy were downregulated in VPS39 silenced cells. And we also found that many genes regulating epigenetic enzymes were differentially expressed in VPS39 silenced cells. So based on this data, we generated the following hypothesis, where reduced VPS39 may affect autophagy, which then regulates metabolism. And we know that metabolism can affect epigenetic enzymes, and this may contribute to epigenetic dysregulation. And since epigenetics is important for cell differentiation, this can then affect expression of key transcription factors regulating myogenesis. And it may affect cell cycle and cell proliferation and apoptosis, which could in the end affect insulin resistance and muscle quality and contribute to type 2 diabetes. So based on the array data I just presented, we generated this hypothesis, and then we went on and tested this. So the first thing we did was to analyze autophagy in VPS-silenced human myoblasts. And as you can see in this figure, the purple is the knockdown and the gray is the control. And you can see the markers for autophagy were clearly altered in VPS39 silenced cells. And that was at baseline. We also studied autophagic flux, and here's just described how you do this. And when we did this in the VPS39 silent cells, we found that there was changes in the autophagic flux when a VPS39 was silenced compared to control. We went on and we tested then if VPS39 silencing impairs insulin-stimulated signaling in muscle cells, and we found alterations in AKT phosphorylation, in TBC1D4 phosphorylation, and nominal effect on the GSK3 phosphorylation, supporting that the silencing also affects insulin's capacity to signaling downstream, and probably also both glycogen synthesis and glucose uptake. We then pursued, because we know that changes in metabolism can affect epigenetics through alterations in activity of epigenetic enzymes. So we both measured activity of epigenetic enzymes, and for those we couldn't measure the activity, we measured the protein levels. And this is quite messy, but the summary is that we had alterations in activity of enzymes regulating both histone modifications and DNA methylation. So we then also measured the expression of key transcription factors that are known to regulate the differentiation of myoblasts into myotubes. So the MyoD and MyoG, and also some of the MEFs. And we found that silencing of VPS39 resulted in reduced expression of several of these key transcription factors that drives differentiation of myoblasts. And then these were all in vitro experiments. We also wanted to see if we silence VPS39 in mice, if this affects the muscle function, and the homozygous VPS39 knockdown is embryonically lethal. So we studied the heterozygous mouse, and we found that when we silence VPS39, these mice had higher glucose during an oral glucose tolerance test. We also used tracers to see if the direct glucose uptake in muscle was affected, and we found reduced glucose uptake in the knockdown mice. We also used array data on the muscle from these mice and found that there was differential expression of genes involved in epigenetics, in autophagy, and in skeletal muscle function. And finally, we went back to the muscle cells from individuals taken from diabetes and controls to see if some of these changes we found when we silence VPS39 also can be seen in muscle cells from diabetics versus controls, since I showed in some of the first slides of this paper that these individuals had lower VPS39. And we found that muscle cells from individuals with type 2 diabetes indeed have some alterations in markers for autophagy. There are some alterations in epigenetic enzymes in these cells. And going back to the expression data from these individuals, this was important that during differentiation of myoblasts into myotubes, it is important that you have alterations in going from glycolysis into using amino acids and fat. And that happens in the control cells, but to a less extent in cells from diabetic versus controls, supporting that there is important changes in these cells. So to summarize this paper, we have established that VPS39 is reduced in myoblasts and myotubes from individuals with type 2 diabetes. This seemed to affect their autophagy, and we found dysregulation of metabolism. We found dysregulation of epigenetic enzymes as well as DNA methylation. And we found alterations in key transcription factors and increased expression of proliferative genes. And together we believe that this then affect insulin resistance and the quality of muscle in type 2 diabetes. So overall, to summarize this talk, we believe that the epigenome plays an important role both in obesity and in type 2 diabetes, and that several of the non-genetic risk factors and genetics affect this epigenome. And we believe that epigenetic biomarkers may be further developed and used for precision medicine in type 2 diabetes. And we believe that some of the data that I presented may be new therapeutic targets for diabetes. And finally, I want to acknowledge members of my group, our funding, our collaborators, and in particular members from Latvia, where Monta and Ilsa and Yanis helped us to establish the European replication cohort, and where we analyze methylation in OptiMed. And finally, I want to thank you all for listening.
Thank you very much, Charlotte. It was very interesting, especially this for me, this VPS39 gene study. So much experiments done, very functional.
Oh, sorry, I didn't see that. I missed my whole. You should have told me.
Sorry?
You should have told me I didn't have my camera on. Sorry.
Well, I was occupied with watching the slides.
Yeah. No, I was a little bit confused with the time. So we had a group meeting before, so I was a bit stressed coming in. Sorry.
It's okay, and it was almost perfectly in time. So thank you very much. And yeah, the last presentation in this first part is from Professor Jānis Kloviņš. Are you ready?
Yeah, sure. So I guess maybe Charlotte can stop sharing her presentation, and then I can put on mine. Okay,
yes.
Thank you.
Yeah, thank you.